There you go.
Welcome everyone to this, our first live and in-person Center for Values in Medicine Settings and Technology event in a little over 2 years. I'm glad to have you all here. I'm very excited about our talk today. My name is Matt Brown. I'm the director of the Center for Values in Medicine, Science, and Technology. I want to just acknowledge a few people who are with us today. Magda Groman, who's the Associate Director of the Center, who does a lot of the crucial legwork for what we're doing, is here. Dennis Kratz is here, the founder of the Center. So welcome, Dennis. Marvin and Kathy Stone, generous supporters of the Center, are here. And two of our research fellows are here as well. Gregorio and Arlen are here, so welcome to you. And welcome also to all of our audience who are tuning in via YouTube, our first YouTube live stream lecture as well. So we're breaking some new ground today. It's my great pleasure to welcome our speaker today, Dr. Scott Podolski, who is a primary care physician at Massachusetts General Hospital, as well as professor of global health and social medicine at Harvard Medical School, and also director of the Center for the History of Medicine at the Francis A. Countway Library of Medicine, also at Harvard. Dr. Podolski is the author of many books and articles on the history of medicine, especially several important works on the history of antibiotics, and on today's topic, the history of clinical trials. We're very excited to hear about this topic today. And I will scoot things over to his slide deck. And let's welcome Dr. Podolski up to...
Thanks so much for that introduction, Matt. Can you all hear me okay? Great, I want to really thank, this was just a lovely 2 days here. I want to thank Magda, Arlen, and Mario for being fantastic hosts, and Dennis and Marvin and Kathy for making this all happen, and everyone who showed up in person. Thanks. It's my first talk in person this afternoon for 2 years as well, so it was a fun time. So, for those who were there at this afternoon conference, we talked about this course that we teach at Harvard Medical School on social medicine. We do a session on therapeutic efficacy and how we know something first. And we'll ask the students, speak a little louder? Great. And some will say, well, based on mechanistic principles, how it works in vitro or on paper. And some will say, well, I took the med, I got better, therefore it works. We'll call that a testimonial. And others will point to more empirical notions, manifested sort of today in the randomized control trial. And this was often this sort of academic discourse that we would have. But really, in recent years, it became front page news, nighttime talk show news. We saw this especially with respect to COVID. Right? We saw the advent of hydroxychloroquine in the very first months of the COVID pandemic. And it became immediately politicized. You had, for instance, Laura Abraham saying, the drug's a game changer in the fight against coronavirus. Anyone who questioned the drug's efficacy, she said, was in total denial. I love everybody. I love the medical profession. But they want a double-blind controlled study on whether the sky is blue. Of course, within weeks, that study was retracted. Based on whether like patients were being compared with like patients in the control group, based on the endpoints that were chosen. And these are all part of a long history of debate over how RCT should be done. And still, President Trump saying of hydroxychloroquine, I think it's good. I've heard a lot of good stories, and it's not good. I'll tell you right. It's easy to pick on Trump and say that in this case the RCT was being politicized. But I'm going to argue today that the RCT and clinical trials have always involved the interface of the methodological, the social, the cultural, the regulatory, political, and the economic. And so we're going to go through this today, about 45 minutes. I'll go to a brief overview of the historiography, and I'll have the slides so you can read the site at the ledger. And then break down the methodological evolution of the RCT. These issues of fair allocation, right? How do you make sure you're comparing like with like in your active versus your control groups? Then we'll look at the history of assessment, and particularly unbiased assessment, both by the subjects or patients, how they report symptoms, and by the researchers and how they evaluate those symptoms. That's the history of blinding. And looking at how just because you have this methodology in place doesn't mean it's going to get implemented. So we're going to look at the combined regulatory political implementation of the randomized control trial in the 20th century before looking at the remaining problems that are found, that exist, even after you have this seemingly gold standard increase. So, just a brief overview of historiography. These are some of the earliest articles on the history of randomized control trials, and I don't think it's an accident they came out in 1959 and 1982. And so we hear about, this is just some foreshadowing, 1959 was the advent of our Harrison amendments, hearings, and this is really a inflection point when the controlled clinical trial and the RCT becomes central to thinking about how to adjudicate drugs. In 1982, we'll hear about the RCT being called the gold standard for the first time. Those articles were followed by a series of books. So in 1995, J. Boston Matthews' Quantification and Question of Medical Certainty looked at the advent of the numerical method. Again, I'll be talking about all this in the mid-19th century and biometrics and really the rise of empirical studies in science. Harry Marx's Progress of Experiment is really the Bible. They're looking at how the RCT gets used in the 20th century as a way to tame the therapeutic marketplace in particular. And then Carpenter's really magisterial book on reputation and power is on the history of the FDA. And it's linked regulatory power, it's keeping power from drugs, and it's conceptual power around the RCT and the role it's played and still plays today. And so certainly recommend all of these. Earlier today, we heard about the notion of how do you include multiple groups in RCTs, and so sociologists and anthropologists have chimed in as well. So Stephen Epstein's In Pure Science looked at how AIDS activists, particularly in the 1980s, really started shaping how the FDA would adjudicate the drugs and shift, and how activists can actually shift some of the endpoints that are getting chosen in those trials. Gene Daly's evidence-based medicine looked at the rise of sort of skepticism in the 70s, in the 80s, that led to entities like the Cochrane collaboration and the rise of quote-unquote evidence-based medicine that's being juxtaposed to, say, eminence-based medicine. Later on, in more recent years, we had sociologists and anthropologists like Adriana Petrina looking at the rise of contract research organizations. Right? How are trials being done today, and where are they being done? Many of these are being offshored by groups that simply run trials, which has its own implications for the kinds of knowledge that gets generated. Whereas Ambrosio Peter Keating looked at the evolution of clinical protocols, especially in oncology. It's very kind of understanding in the audience here. And these lineages of protocols that evolved from the 1950s onwards. One of my favorite sites, for anybody interested in this, and this is a sort of conflict of interest, I'm on their editorial board, but this is the online Jameson library. And they have nearly 1,200 original articles related to the history of RCTs on there, another few hundred interpretive articles that have been written by scholars about the history of RCTs. If anybody is interested in this, I strongly recommend just going to Jameson Library. You can find great primary sources and interesting secondary sources as well. And that brings us to James Lynn. This is sort of the start of the story, right? How do we have like compared with Lynn? And does anyone know who James Lynn was? Right, so James Lynn was a Scottish physician. He was a British naval surgeon, and he was on the ship. He was a ship's doctor in mid-18th century Britain. And his famous quote is that on the 20th of May, 1747, he had 12 patients with scurvy on board the Salisbury Sea. Their cases were as similar as I could have them, and you have this like being compared with like. They had the same symptoms, they were in the same location, they had the same common diet, and he divided them into 6 groups of 2, each getting a different additive. Famously, quote, 2 had each 2 oranges and 1 lemon given them every day. They continued with 6 days under this course, having consumed the quantity that could be spared. The consequence was that the most sudden and visible effects were perceived from the use of the oranges and lemons. One of the 2 sailors leading this regimen recovered. He was pronounced fit for duty. The other was so well that he was appointed nurse to the rest of the sick. And this study becomes the touchstone for the controlled clinical trial. This is why Jameson is famous in this respect. This is happening institutionally by the end of the 18th century, early 19th century. You start seeing some factors in place that promote more numerical evaluation. Right? So, for instance, post-revolutionary France, you have the advent of major hospitals in Paris. And patients concentrated in one space. So you can have people like Charles Alexander Louis, who actually called numerical method. They're actually comparing remedies to either one another or to those who didn't get them. And famously, for instance, I worked on the history of pneumonia. He was comparing patients who were bled. So the treatment for pneumonia at that time was phlebotomy. It actually cut open the vein. And if you're skeptical about this, and there was a great deal of skepticism by Louis, you can evaluate this. He found that those who were bled early in the course of the disease did no better than those who were bled very late in the disease, really sort of discrediting the disease per se. Now, a whole cohort of American physicians went over to Paris to learn under the likes of Dr. Lee. And they came back really advocating for this numerical method in 19th century America. One of these was Alfred Holmes at Harvard. And that's him, second to the left on the screen. And he said in 1844, at the start of his career, the true question for the jury is not, do hospital or other physicians try experiments? For strictly speaking, every administration of a remedy is an experiment. But do they study diligently the claims of all new and old methods? And do they know how to select those which offer the best chance of proving useful? This was an aspiration to a rational therapeutics. The problem is that in the mid-19th century America, there was no infrastructure to do these studies. There were very few great hospitals. Now this is what's happening in Paris. So that by the end of his career, in 1882, Holmes will more cynically lament, the numerical system can teach a wise, honest, and diligent man a great deal, and can make a foolish, dishonest, careless man a greater fool, impostor, and blunderer than nature ever intended him to be. And in fact, some of the earliest studies that really tried to implement some form of clinical, quantitative evaluation weren't what we would think of as proto-randomized control trials, but they were what we called collective investigation. So, for instance, by the end of the 19th century, at the advent of germ theory and the advent of immunotherapy, so one of the first great therapies, actually, was what would garner the first Nobel Prize in medicine in 1901, was for the use of serum therapy for diphtheria. And the American Pediatric Society tried to figure out, does this work? And figure out how can they eliminate my bias versus Dennis's bias versus Martin's bias. They said, everyone across the country, please mail in your results. We had 613 physicians from 114 cities across the country mail in their results, and they found that serotherapy, when used, yielded about 13% mortality rate, which was much less than they expected, say, 25 to 30% mortality rate that you would otherwise have had. But by this time, others were starting to implement things that are more now to think of today as controlled clinical trials. And this notion of alternation. How can we compare like with like to remove confounding variables? So one of the first of these was done by a man named Alexander Wasassier Hamilton. This is a different Alexander Hamilton. He was another surgeon during the Peninsula Wars with Napoleon at the start of the 19th century. And he had about 366 patients with various kinds of wounds. And he said, let's have 2/3 of them not get bled, 1/3 of them get bled, and let's compare results. And he alternated the patients, right? So A gets it, B gets it, C doesn't get it. B gets it, E gets it, F doesn't get it. And he found that those who were not bled did about 10 times better. And again, you can see this skepticism right around bleeding. And the other big object of skepticism during the 19th century was homeopathy. So similarly, Thomas Graham Balfour was a superintendent of an asylum in Britain in 1854. And belladonna was homeopathic. And he said, I'm going to assess whether homeopathy actually works to prevent scarlet fever related to cataclysm. So he says there are 150 boys who are tolerably satisfactory evidence that they didn't have scarlet fever. I divide them into two sections, taking them alternately from the list to prevent the imputation of selection. As we hear the word today like selection bias, already in 1854 he's mentioning basically de facto selection bias. And he alternates them. Boy A gets it, boy B doesn't get it. Boy C gets it, boy D doesn't get it. So the first section, 76 of them, I gave belladonna. To the second, I gave none. The result was that 2 in each section were attacked by the disease. The numbers are too small to justify induction less than the prophylactic power of belladonna. And the observation is good, because it shows how apt we are being misled by in perfect observation. Had I given the remedy in all the boys, I should have probably attributed to it the cessation of the epidemic. Right, so this is already a pretty sophisticated explanation for the necessity of comparing life with life. By the 19th century, when it starts to really, near the 19th century, when this really takes off, though, there are a number of factors in place. Again, you have an increasing rise of hospitals in the U.S. and Britain. You have the rise of germ theory and a lot of remedies meant to reduce bacterial infections. We also have a lot of junk. The real flooding of the marketplace with what are considered to be patent remedies. So, for instance, Vin Mariani, I don't know if you've ever heard of this, is a combination of cola and alcohol, and at some points cocaine. And you had this advocated by some testimonials, whether it's Pope Leo XIII on the right, who advocated this, and was widely pictured with respect to this, or the 8,500 doctors who use Mariani wine. Right, so there was this flooding of the marketplace with a lot of junk. And not just in the popular literature, but this is Boston Surgical Medical Journal, which is the New England Journal of Medicine. We had similar types of remedies. So there's this attempt by some to figure out how can we figure out therapeutic needs from chance? How can we achieve a quote-unquote rational therapeutics? And the AMA at this time creates what's called the Council on Pharmacy and Chemistry. Right? So it's at least trying to say we need to figure out a way to adjudicate needs versus chance. And around this time, you start really seeing the advent more and more of what is considered alternate allocation trials. One of the first ones, for many years, they thought the first one was in Copenhagen, by Johannes Feiger. And this again was for diphtheria. And he alternates 484 patients every other day. Patients on Monday get it, patients on Tuesday get it, don't get it, Wednesday gets it, etc. And he finds about a 4-fold improvement in mortality rates with anti-diphtheria and anti-serum. And Ian Chalmers, George J.B. Smith, and Stella Duca and I looked into all issues of JAMA and NEJM and Lancet in the half century between 1898, Irish study, and the 1948 British Earth trial. And in JAMA alone, we found about 130 mentions of these alternate allocation studies. 48 of these were original studies, 80% of them were infectious disease related. So you start seeing this increase in the half century between 1898 and 1948. The one I worked most on, and I think is pretty telling, is Anthropococerotherapy, the treatment for pneumonia. Right, so Rufus Cole at the Rockefeller Hospital in the early 1910s comes up with serotherapy for pneumonia. And he's so convinced that it works, he's ethically feels it's unjustified to do a study. He says, I'm just going to, this works in the lab, it works in rabbits, I'm therefore going to give it to people. And it works. I'm not going to subject anyone to the control group. Then what happens, you have the act of World War I, you have a number of then soldiers in close proximity to one another. It takes to be a good place to test it. And of course, you have the advent of the influenza pandemic. And it's a mess. And the Metropolitan Life Insurance Company loses $23 million in excess death benefits during the influenza pandemic. And most of those patients are dying of secondary pneumonias after the original flu. So we're like, wow, we think we have this remedy that might work. We don't know when the next pandemic is going to happen. We need to figure out whether this stuff works or not. So they actually fund alternate allocation studies in multiple hospitals along the East Coast. Boston City Hospital, Bellevue Hospital, Harlem Hospital, and it's pretty sophisticated. Right? So this is just below studies of antipneumococcal antiserum in these New York hospitals. And you can really see again, since 1928 on the left, those ratio of difference to its errors, that's basically equivalent to a p-value. It's above 2.5, it's about equivalent to p-value of less than 0.05. We have pretty much meta-analysis here. They're combining studies from multiple hospitals. On the right side, you pretty much have post hoc stratification. It's pretty sophisticated, yet still somewhat new. Right? So when he actually gives a report on this to New York Academy of Medicine, the very first, Academy of Sciences, New York Academy of Sciences, the first section of the paper is actually called the control. He needs to justify why we're even doing the control group. But basically he's saying here, I don't want to dismiss a remedy that may have not worked for the first one unless it's really good. All right, so these are happening in the United States. There are several of these popping up by the early 1930s. And around this time, the British Medical Research Council decides to do its own study of antipneumococcal therapy for pneumonia. And their chief person working on this is a guy named Austin Bradford Hill. And he finds something very funny. He says, if you look at this, we're supposed to be alternating it exactly, right? Patient A gets it, patient B doesn't get it, patient C gets it, patient D doesn't get it. And if you do that perfectly, statistically, it's just as good as doing an RCT. Basically, now looking at this, and it's here people are getting zero therapy. Basically, people are cheating. And they're well-intentioned, right? I'm a physician, patient A got it, patient B didn't get it, patient C got it, patient D was sick. I actually think this is going to work. I'm going to give it to him, and I'm going to start over again. And he says, we can't have it this way. So he says, for my next big study, what I'm going to do is I'm not going to do strict alternate allocation. I'm going to do concealed randomized allocation. So that there's no way that anybody will know which one is up next. Right? And that becomes the basis, what's considered to be the first real randomized controlled trial in history. This is the 1948 trial, obstructive license for tuberculosis by the British Research Council. And you can see how it derived from the alternate allocation studies, but with this twist of concealed alternate allocation. Right? So that's like comparing with like. What about unbiased assessment? This is both of patients and of researchers, right? How do we know that we're getting real data? And the notion of blinding patients, right? Blinding meaning making them not know if they're getting a treatment group or the not group, has these origins and skepticism that goes back centuries. Right? This goes back to Ted Capture did this work on exorcism trials in the 16th century. Right? Because it's easy, like Martha goes in here, seeming like possessed by the devil. I'm going to give you this sham, this treatment. Did it really work, or was it my charisma that was doing this? And so you had people doing sham holy water, sham crosses, and really trying to figure out whether this worked or not. And we heard about Montaigne today there during lunch, and he was actually recording these, and he was around. People like Benjamin Franklin were reading Montaigne at the end of the 18th century. And similarly, in France at this time, you had Anton Mesmer. And he was seemingly curing people of their illnesses through this manipulation of animal magnetism, through mesmerism. And again, if you're cynical or at least skeptical, you want to test this. So Franklin and his group, which included Anthropococerotherapy, were doing sham mesmeric trances. They were mesmerizing people through a literal blindfold or through a curtain to see if it worked. And again, they found out that it didn't seem to work. So again, this notion of the need to blind patients to get true responses was prevalent. This got extended again to homeopathy in the mid-19th century. Again, you can see the skepticism. But soon gets extended to conventional pharmacotherapy as well. So in the United States, for instance, angina, which is heart disease, for all the folks who are pre-med here, this notion of a chest pain that's in two blockages. And if you look at the literature, the 20s and 30s and 40s, it's famously a very variable disease. You don't know, and it's seemingly a very suggestible disease. And so Harry Gold, who was at Cornell, was doing these trials of therapies for angina. And he said, some patients, this is the first instance of the use of the word placebo in a trial. He said some patients in the study expressed a definite conviction at times that it was the drug which was responsible for the relief. And this drug was often the lactose placebo, and that some patients insisted on its efficacy, protesting against any suggested change, justifies all the circumspection one can exercise in accepting a patient's judgments in a study of this sort. Right? This is a justification for the need for the placebo group receiving a sham therapy in trials. And it wasn't just for symptoms. People were thinking this would actually impact outcomes for non-symptomatic endpoints as well. So around this time, the late 20s, early 30s, Park Davis had a gold remedy for TB. And Park Davis, along with the health service, ran this whole trial. And they divided people in half, and they used to have a sham group. Here, if you look at the archival records, and this is what Joseph Gabriel says, in order that the psychic influence may be eliminated, the control group shall receive injections of some fluid, such as sterile water. So by this time, we already had this notion we had to blind the research subjects, but the evaluators were also biased. Right? I come into this trial, and I kind of think I know whether I like it or not. I need to be blinded as well, so that I'm not biasing my results as well. Again, this starts off in the 19th century with evaluations of homeopathy, but soon extended to conventional immunotherapy and pharmacotherapy as well. Right, so this is a British report from the early 20th century, but I think it's telling. This was a study of inoculation for whooping cough prevention. And this guy, Dr. Friedman, says, after the children had been inoculated in the way I had described, I constructed statistics from the reports of parents to see what effect, if any, had been produced by the vaccination. For this purpose, the random remarks of parents were reduced to one of 5 following simple statements: much better, better, no change, worse, and much worse. In doing this, quickly found I could not rely on my own impartiality. For instance, if the mother reported the effect that the fits of coughing were more severe but not so frequent, I found the statement tended to appear as better in the case of the vaccinated and worse in the case of the controls. I got the obvious remedy of remaining in ignorance as to whether the child was being treated in a control case until I got the results safely classified by the board. I'm convinced that without these precautions, the investigator will usually find nearly what he expects to find. I mentioned that AMA Council on Pharmacy and Chemistry. And when they were doing these studies, they too were blinding the researchers. They were giving these medicines to treatment. In this case, this was for aspirin versus synthetic versus natural aspirin. To see, they would give them a label or boxes that were labeled A or B. And they looked at the personal equations of different observers, the tendencies of bias, all tend to obscure the significance of reported results. And this personal equation to me is fascinating. Side history. This is usually an astronomical term. So if Magda and I are both at the same stars and charting them, I'll see it one way, she'll see it another way. There's this personal equation. It's sort of, it's a cupcake story, but it has some notion of bias to it. You can see the way it's already peaking during this time. And how do we remove the personal equation and turn us into quote-unquote objective measures? And in fact, as late as 1946, you're seeing, 47, we're seeing these notions of the personal equation in the evaluation of the chest x-ray. That again, that if Marvin and I evaluate the same chest x-ray, he may call it pneumonia, I may call it not pneumonia. And in fact, I may call it pneumonia on one try, and not pneumonia on another try. And in fact, this is right before the British MRC trial of streptomycin. And actually, what was being blinded in that particular kind of trial were the radiologists who were reading the x-rays on those determinants. All right, so by 1948, methodologically, everything's pretty much in place. You've got light compared with light. You've got the infrastructure for unbiased assessment. And you've got Patty Gold basically saying the whole history of therapeutics, especially after there was actually drugs and suggestive symptoms, demonstrates that the birth of one study is quickly reversed by another unless one takes measures to rule out the safe, effective medication of the patient and the unconscious bias of the doctor. The double blind assures us. And this is really the first usage of the term double blind. So it's all in place. You've got statisticians like Austin Bradford Hill and Donald Neal going around trying to pretty much proselytize out the need for such unblinded assessment. And yet, no one's forcing anybody to do this. There's no teeth to this. Right? So when Wilford Ross in the early 50s looks at the medical journals and see what percentage of his trials are actually these gold standard kinds of trials, only about 27% fit that bill. Right? Again, because no one is forcing them to do this. No one's certainly forcing the pharmaceutical industry to do this. There's no teeth to this yet. So now we get into the regulatory history of the RCT. And again, to do this, we need to have a quick overview of the history of the FDA. Right, so the FDA is formed in 1906. This is simply to, in the context of most drugs, either having cocaine or alcohol or heroin, it's basically mandated that drugs contain what their labels say they will contain. Things go along for several decades. You have the advent of powerful drugs like the sulfur drugs in the mid-30s. Sulfanilamide is excellent drug. We have strep throat. It tastes horrible. The chief chemist that kids will take, the chief chemist of the Mexico Corporation says, I know how to make this powder and so they have glycol. It turns out to be the main ingredient of antifreeze. 100-something children. The chief chemist kills himself. This pretty old extra food drug comes out back in 1938. It's safe. Not necessarily efficacious. Evidence of state. Sale. Efficacy is sort of implicit here, but it's not explicit. Right? This is sort of the prelude to sort of the therapeutic jungle of this called. It's a massive corporation of drugs that are having capital relations. But this is usually the active antihypertensives, steroids, antipsychotics, tranquilizers, and especially my favorite topic, antibiotics, which play a key role in the history of drug regulation during this time of the advent of the RCT. And so penicillin comes out during World War II, streptomycin comes out in the latter half of the 1940s. By the late 40s, early 50s, you have the advent of broad spectrum antibiotics. Pfizer's terramycin, Arthur's chromacin, and Lilly's dorylamycin. And these are massively profitable drugs, right? So you can see the literal change in scale of drug sales there. These are, right? So when Anthony Macopoulos here, I was talking about the $1 million a year drug, dorylamycin is a $60 million a year drug. Which is interesting, is an $80 million a year drug. And he had this arms race. So given bugs like staphylococcus were resistant to penicillin, sensitive to the broad spectrum antibiotics. And you found bugs that were okay, we're in this arms race. We know what we're going to do. We're going to come up with another class of drugs, but fixed dose combination antibiotics. We're going to take one of our good drugs, we're going to mix it with something coming through the pipeline, like Jameson, and we're going to call this fixed dose combination synergistic antibiotic. And at the 1956 International Symposium on Antibiotics, the head of the FDA, Division of Antibiotics, says, we're now in this third era of antibiotics. The first era was the nanospectrum drugs, penicillin. The second one was the broad-spectrum drugs, and now we're in this third era. You guys at Max Finland, Harry Dowling, Max Finland was at Harvard, Harry Dowling was announced the University of Illinois, and then Finland's first fellow. They're very skeptical. They're saying, this is job. Not only can such antibiotics be synergistic, but they can be antagonistic with one another, and moreover, you can't show that any given one works or any given bug unless you go through the testing. So the notion of selling this, right, that selling this as a remedy to the current doc like me, to just give to all your patients regardless of what they have, this is the admin style over substance. This is marketing over science. They have to do something about it. So Dowling uses this in a very famous speech. He'll twist a couple of to say, we're paying this dystopia. He says, well, if we keep doing this, like we're advertising as how we have to treat doctors, then we're going to be no different from those who are advertising soaps, toothpaste, cigarettes, whiskey. Right? And with the inevitable disillusionment that comes from the failure of each useless modification of a advanced pharmaceutical industry will lose its prestige, and with this will lose its financial backing. It will fall, and the medical profession will be dragged down. This was a dystopia around national therapeutics. Finland takes a somewhat more practical course. He says that we've got to counter this with something. Right? Because we basically say, well, we have testimonies. We have a drug that's Jameson. We give it to someone like me. I'll test them. Here's 100 patients with pharyngitis, sore throat. They all get better. Great. Pfizer will use that study. They'll quote it in their publication. And that is a testimony. Right? It says the admonition to defer acceptance of claims to the manufacturers until they're confirmed with reliable and unbiased reports from other laboratories in support of controlled clinical trials rather than mere testimonials. They've been completely submerged in the deluge of advertising material that follows. So he juxtaposes the testimonial with the controlled clinical trial. When he invokes a testimonial, he's reaching back to the 19th century patent drugs. He's basically saying the ethical pharmaceutical industry is behaving no differently than the patent remedies of the 19th century. This could have been just a totally different professional discussion, right? It was for a while. And he and his colleagues are fighting the journals about this. It gets taken up by the editor-in-chief of the the science editor of the Saturday Review, John Lear, who writes this article of doctors and antibiotics taking the miracle out of miracle drugs. And that's the magazine I'm holding up, one of those pictures that was used for the promotion today. And this happens in early 1959, just as Senator S.C. Kefauver is beginning his hearings on the pharmaceutical industry. So Kefauver was a liberal senator from Tennessee. He'd taken on a mob in the early 1950s. Anyone seen Godfather 2? And Senator Kefauver at the beginning of the movie. And this in the 50s had been a wonder era for the pharmaceutical industry. These drugs were expensive. And it seemed at times to be portrayed with monopolistic pricing. So Kefauver was originally going to start these hearings around monopolistic pricing, around patents. And when he reads Steve Spears' article and sniffs Steve Spears' article, he said, we have to start writing about drug. We have to start having hearings about drug marketing. And in fact, the inability of the FDA to explicitly adjudicate drug efficacy. That becomes a key subtext of the hearings. These hearings go on for 3 years. There are bills that get promoted for having to regulate the drug industry. They fall off the table. And then by mid-1962, we had the advent of the live line, right? The crisis around the live line. And babies being born without lives, with familia, in that context. And President Kennedy and Congress said they've got to pass something. So while all the original provisions around patents, monopolistic pricing, they'll all fall out of the bill. This mandate, the drugs be proven efficacious, based on substantial proof of efficacy, based on adequate and well-controlled investigations, including clinical investigations, is written into law. And this really becomes the underpinning of the entire phase system of drug evaluation really after this time. There's 2 big questions that are left from this. A, what do you mean by adequate and well-controlled? And B, what do you do with like the 4,000 or so drugs that have been on the market from 1938 to 1962? So in 1966, the head of the FDA, James Geiger,
reaches out to the National Research Council to form a bunch of 30 or so expert panels. It's called the Drug Efficacy Study. And they're studying all existing drugs in the marketplace to see if they meet these criteria. 5 of them are devoted to antibiotics. And by the end of 1968, they all find that all those fixed-dose combination antibiotics, like cipralizumab, should be taken off the marketplace. And at this point, the chief issues is no longer Pfizer cipralizumab, but Upjohn's Canalba's combination of novobiocin and tetracycline is a $30 million a year drug. And these panels find that the drug seems no better than tetracycline alone, which should be taken off the market. Upjohn sues. They sue the FDA. They go, they have a bunch of doctors write in to the FDA saying, how dare you take away our therapeutic autonomy. Upjohn says, you're changing the rules in the middle of the game here. This drug was put on the marketplace in 1957. It's still a good drug. And it goes through the courts throughout 1969. And again, it sounds, in a way that really foreshadows what Trump was saying. The Upjohn lawyers say, because basically the FDA says, all you have are in vitro data, testimonials, we don't have real clinical trials. And they say, we don't have just testimonials. We have a very extensive compilation of studies, in vitro, animal, human, coupled with very substantial clinical experience. I think taking that together, it's a factor. And in the setting, the head of the FDA, Herbert Lay, is forced to define what he means by a well-controlled study. Basically, it's a double-blinded, randomized, controlled, placebo-controlled study. And he says, look, this is the gold standard. And if you don't have, if you're not doing it this way, you better not really do these. Winds its way all the way to the Supreme Court, which finds ultimately in favor of the FDA. This was a pivotal moment in 1969, 1970, when empowering the FDA to shape the therapeutic marketplace, and really becomes, in my opinion, the apotheosis of the advent of the randomized controlled trials at the top of the methodological hierarchy, where it's really remained to this point. By this time, you have people like Thomas Chalmers here, Michael Condrey in the UK, began trying to combat evidence-based medicine with evidence-based medicine, really saying that all remedies should be evaluated through rigorous studies. By 1982, Alan Feinstein calls the randomized controlled trial the gold standard. By 1991, you have the notion of evidence-based medicine, which persisted this time. And the RCT sort of near the top of the case studies down here, retrospective studies here, RCTs here, and then the meta-analyses and systematic reviews built on them at the top of the methodological hierarchy, which in some ways has remained to this time. But there's so many remaining persisting gaps and challenges to this time. One, as it always has been ethical challenges. Right? So if you think that you have a remedy that really works, when are you comfortable subjecting anyone to a controlled trial to be in the placebo group? And really, if you look at the history of the end of RCTs, it's really about often outpatient studies. It wasn't that they were so methodologically sophisticated, it's that people really weren't so eager to subject people to a control group. And we saw this with the group is called and Anthropococerotherapy here. And we see this in the 30s with respect to convalescent serum, at the same time that we heard about with COVID. This is being promoted for polio in the early 30s. And this is from the New Journal of Medicine. And you can see again this discomfort of subjecting anybody to being in the control group. With the wave of infantile paralysis that has come rolling up in the South, there's a new surgical alarm. And as a consequence, a revival of fervent hope, some means may be found to shield our young against afflictions. Parents are not to be persuaded or urged to volunteer their children for the trial of new biologic agents. That's different from today. They demanded that. It was this parents' plea that instructed the carefully planned investigations of the value of convalescent serum. Needs for a careful appraisal were easy to devise, impossible to carry out. The main difficulty encountered was the inability of special investigators to withhold this promising agent from any stricken trial. The plea for serum was too impassioned to be resisted. Our sentiment overruled our reason. And these concerns will persist for decades, and it still persists today. So in oncology, again, and I'm sure Dr. Stone can attest to this, when it's had it seems to be a useful remedy, it's hard to subject someone to be in the control group. This is from Baylor, MD Anderson in Houston, but here in Texas, where he had Evan Gahan and Neil Frerick saying in 1974, sometimes the initial clinical studies suggest new therapy is much more effective than conventional therapy. In this circumstance, clinical investigator would not be fulfilling his ethical responsibility to his patients. He'd be doing the randomized comparative trial instead of administering the therapy to consecutive patients. And you had the eventual outcome of the notions of clinical equipoise. Right? When are we about 50-50 as we decided to allocate someone to a control group that comes in subsequent decades? It remains a tricky issue to this day. And what about, are there aspects of medicine that aren't as easily amenable to the double-blinded randomized control trial? Sure. I mean, doing psychiatric interventions can be hard to apply to RCTs. And certainly surgery has its own history in this respect, which is a complicated history, right? So, for instance, in the 1950s, there was a therapy for angina called internal mammary artery ligation, where you would tie off my mammary arteries and somehow shine blood to my heart. That would seemingly get me better. This took off in Italy. It seemed to be doing really wonderfully. It was created in this country. Some people were doing the studies, doing the physiology, saying, this doesn't seem to make sense. I don't think it should work. And they do a blind study of internal mammary artery ligation, where some people would actually get the artery ligated, others they would just make a little incision, close up, and see how they did. And it turns out everyone did just as well in both groups. In that setting, Henry Beecher, who we mentioned earlier today, wrote an article basically saying every single new surgery should be proven efficacious through a double-blinded, sham-controlled study, right? Which is saying a lot, because certainly, when we think about the advent of coronary artery bypass surgery, which entails a full sternotomy that happens in the subsequent decade, that clearly is not the case, right? It does come in up though again in Texas in the 1990s. Early 2000 at Baylor, where they actually do a sham study for arthroscopic knee surgery. Like people coming in with knee pain, and eventual models that we all get arthroscopy and seem to walk out feeling a little bit better. They said, yeah, something's a little funny about this. They did a sham controlled study, and they found groups who received the intervention didn't do any better than groups who just had massive incision and anything closed up. But it's a tricky calculus of when to justify to put people in a control group, sham control group, in a surgical study, for those generating knowledge. And so these are considerations still play a role to this time. The second issue that reigns to this point are the contested results. Right? And this could be issues of quote-unquote internal validity. Was the light truly comparative light? What were the outcomes that were chosen? Right? So we saw this with respect to COVID. Is it nasal clearance or is it some kind of mortality standpoint? We saw this with the new Biogen drug, Adelhem, for Alzheimer's disease. Is clearance of beta amyloid sufficient to be an endpoint versus actually some type of clinical subjective outcome? There are questions of external validity, generalizability. Are the people in this particular study applicable to my patients? And that's only even more vulnerable in surgical studies. We can say, well, those surgeons aren't as good as my surgeons or my team. That team isn't as good as my team. So these still pertain to this point. Sometimes you can have a study which seems to be quite good, but it has a limited impact. There's limited uptake, whether by clinicians or perhaps by patients. And this may be shaped by, again, who's giving the information to those clinicians, right? So in this case, this is a study that's showing the impact of old thiazides that were cheap. No one's exactly promoting them, so they weren't being taken up in the way that had been done by a pharmaceutical company. And certainly with respect to COVID today, we can see vaccines which seem to be proved efficacious through good trials, still not being taken up by all patients for a variety of structural and cultural reasons. And finally, and perhaps in some ways most importantly, there's a production bias, right? About what domains of knowledge are being generated through randomized controlled trials. And this dates back in some ways to the Kefauver-Harris amendments themselves, right? Substantial proof of efficacy based on adequate and well-controlled investigations, including clinical investigations, the experts qualified by scientific training, experience, evaluate the effectiveness of the drug involved. Suddenly, it became really hard and expensive to do a trial. Right? It used to be, again, if you were just given to me some doctor to try 100 consecutive patients, that's not so expensive. But to do a good study, that takes real investment. And Laura Bethwell did the work in here we used in our article. Again, who had the bandwidth, who had the infrastructure and the money to do these trials early on? It was usually government, whether it was the British Medical Research Council or the US Public Health Service. But certainly in recent decades, it's been drug or device manufacturers. It's been the pharmaceutical industry. And again, in recent years, a lot of this has been outsourced through contract research organizations. Again, this doesn't mean that these can't be good studies, but certainly the questions that get asked and the answers that are provided are being shaped by the money used to be able to provide, to fund those studies. And that greatly shapes the actual agents that are being investigated here. So, that was a lot. But on the one hand, as you can see from this, all this, while we have our methodological evolution, this is all being shaped by social, political, economic, regulatory forces, both then and now, and still can be highly contested as well. Yet at the same time, the RCT has certainly evolved at the end of its long lineage of attempts, and it's a very fluid category to this time, right? But it certainly has been considered to be central to having some form of a rational basis on which we can stand and try and adjudicate what works in medicine. When we're left back again, I'll give the final quote to Oliver Holmes again. That experience had been, time immemorial, pouring its flowing treasures into buckets full of holes. At the existing rate of supply and leakage, it would never be filled. Nothing would ever be settled in medicine. The cases thoroughly recorded, mathematically analyzed, would always be available for future use. And when accumulated in sufficient number, would lead to results which would be trustworthy and will of the science. Thank you.
Questions? Yes, Marvin.
Scott, that was a marvelous talk. One of the things that I think we have to cope with pretty often, and this is particularly true in oncology, although not restricted to oncology, and that is the conflict of interest issues where we have multiple authors of a paper, even in the New England Journal, which alleges to show a significant p-value difference between whatever they're testing and control. But they're all being paid by the company that makes the drug that's being tested. How do we get around that?
Yeah, I mean, it's an important point, right, in this ecosystem of generating trials. And, on the far end of one scale, you can have this notion of truly a core process where you're just trying to generate patients. There's certainly data that trials funded by industry may be biased more than trials that are not biased. You can have ghostwriters, you can have entire corruption. I don't know what percentage of trials are that terrible, but some are. And then, we try to have this history of these kinds of protections in place around binding researchers to the control group or placebo group that try and prevent bias otherwise. At the very least, it's shaping which trials get done. In the worst cases, I think you're right, and perhaps with some CROs it's possible. And you may have a better sense than I do, oncology trials. What percentage do you think are really that far out to the extreme?
Dr. Podolski, if you take that the next step, those pharmaceutical companies are now paying for expedited approvals, which becomes, it just feeds onto that same thing of how do you know when things, I mean, just that last Alzheimer's drug is a perfect example. How do you protect your public, and how do you maintain an ethical standards when the person who's asking for the approval is paying you to do it?
Yeah, and this tension, I mean, it's not so that it's easy to critique the pharmaceutical industry. There's some great drugs, and I'm thrilled we're boosted. Having said that, there has been this tension at the FDA of being damned if it lets bad drugs on the market, and damned if it's too slow letting good drugs on the market. And it's this where you draw the line of wanting to have this expedited approval to get a new drug on the marketplace to fill some need, versus that becomes a window to let shoddy studies get through. And that is the history of this tension that since from Kiefer onward. And like you're saying, what do you do when the pharmaceutical industry is also funding the FDA? Why can't we just have the FDA funded independently?
And if you add on to that the fact that the federal government is paying them to bring on these drugs, themselves. I mean, a great deal of the funding that they then allocate out to the people who are doing the clinical trials comes directly, it comes through them, but it comes from NIH.
Yeah, yeah.
And so we're paying for the drugs, and they are reaping the benefit of the profits from the drug.
Sure.
And unfortunately, when large institutions have their incubators and they are producing the science before the drug becomes approved, and they sell it to that rich Boston company. I'm not talking about one, but there's lots of pharmaceutical companies in Boston. And they make no allowance for the fact that a portion of the public will not be able to afford it. So the university makes money on it. The pharmaceutical, assuming they get approval, makes money on it. The money was maybe allocated partly from the federal government, and yet poor people, no one bothers to negotiate in that process for people who cannot afford the drug, even with their programs that they talk about for assistance programs. It doesn't cover anywhere near the people that really need to.
Yeah, I mean, if you look at this history, there's two tensions, right? There's the too lenient, too stringent tension, and there's this innovation access tension, right? And so, again, you can see it in the key hearings themselves, where you had the critics saying, this monopolistic pricing, this is raising prices, you're impeding access. They would say, well, great, how many great folks have come out of the Soviet Union? This is funding, we need this for innovation to incentivize innovation. Which is still what Farmer says today. And you still have that tension between those two goals.
So I wanted to ask, and this is drawn from my relatively limited experience, but when I was in lab, I noticed this push towards using more complex and more accurate statistical models with each increasing study. And what I wanted to ask about is, with this push towards more complicated models that aren't better predictors, but are also harder to implement, how do you avoid researchers assuming that the numbers aren't right because they did the model wrong and adjusting to fix the numbers rather than it actually being a problem with their data itself?
Yeah, I mean, right, even the best intentioned studies are relying on certain parameters which may or may not be correct. So while you have this basic framework of what constitutes an RCT, it's clearly an evolution. Whether it's being for RCTs of therapeutics or association studies, or there's our Center for Bioinformatics at Harvard just did this whole study where there's these looking at association studies and how changing parameter study can come up with entirely different outcomes. Like you're saying, it's also kind of like a vibration study where they're playing with those parameters. So, yeah, I mean, it's what splits a lot of contested studies, and some studies that are not complicated.
I have a question from the chat. What role has the patient community research advocates or activists played in the development of clinical trials?
Well, most famously, this happens with AIDS activists. That was the impure science part of the program that I mentioned with Jameson, where in the 1980s, when you had, in the 90s, you had, before they added the therapy, you had, as I say, we can't wait for these long studies using, let's say, mortality outcome as our answer. By that time, we're all going to be dead. And so there's all these negotiations about how you can change the parameters of studies. So, for instance, this is the advent, played a role in the advent of biomarkers. Why don't we use CD4 counts as a surrogate marker for mortality data? And that's had wonderful aspects to it, the acceleration of drug approval, but it has this tension to help whether surrogate markers are truly the outcomes that one should care about. And we saw this again with Allocation. We'll see this with other studies. So patients have certainly played a key role, especially in the last 30, 40 years, often in trying to accelerate drug trials. Other times where they could be astroturfed by pharma, it's for them as well. I think that's the most famous instance in that respect.
Yeah.
What made you interested in this sort of research?
Yeah, I'll tell you. And I bring this up, especially with the students, to say that once you give them a question, it leads to other questions, which then have their own. For me, it's been a 20-year kind of process. So that I mentioned to you earlier. I was an evolutionary biology, partly within history of medicine. As an undergrad, I was very interested in the tempo and mode of therapy of evolutionary change, and my idol was this guy Stephen Jay Gould. Anyway, I was starting my pneumonia book on the history of treatment of pneumonia for anesthesia. I was really interested in the tempo and mode of therapeutic change. How you went from stereotherapy to sulfa drugs, and what led to shifts. Anyway, as I was doing that, I was tracing the history of stereotherapy. I was like, wow, these people are really wrestling with whether this stuff works or not. Right? So we have this whole question of the history of changing notions of what constitutes efficacy. And I went backwards, to those trials of stereotherapy in the 20s and 30s, and then tracing that forward, just got into the broader history of controlled clinical trials. So really, it started off with just looking at tempo mode of therapeutic change, and once you're in the literature, you'll see other questions that start to pique your interest. That was it. I will say during that entire time, though, both the original one was while I was in the hospital, watching as a resident, watching one therapy come in and another, which was really inspiring what I was doing. This question of efficacy was also happening as I was in the clinic. With engaging with patients about, doc, does this work? How do we know it works? So clearly a time when what patients are voicing in the clinic was also shaping what I was thinking about as a historian.
Scott, we talked briefly last night about Paul Farmer. Would you just mention some of his contributions?
Oh, sure. Mark was asking about Paul Farmer, whom I mentioned at lunch, and if folks haven't don't know about Paul, just a remarkable person. Was an anthropologist and humanitarian. It was really, really grounded in notions of social justice and equity. That notions of pragmatic solidarity that really anybody around the world deserves the same care that we would get in this room, right? The least privileged deserve the same care that the most privileged do. We should be really thinking of the structural factors that impede that. It's often been easy to ascribe, say, cultural factors to why people aren't choosing a certain remedy. One should always be thinking of the structural factors: poverty, racism, gender violence, etc. Structural violence that's impeding individuals' agency. It's funny, I think Paul at times would contest the necessity of a randomized control trial. I think sometimes you can say some of these things are obvious. Why do I have to prove that giving somebody, he would say, not only do I give my patient treatment for TB, but why am I giving food and money that's going to improve outcomes? Are you really telling me I need to subject that to an RCT? Isn't it obvious that someone who's well-nourished will do better than someone who's not well-nourished? So I think Paul had a complex relationship with RCT, but his relationship was always extraordinarily grounded in notions of equity and social justice, and really true accompaniment, really being there with his patients. He's a phenomenal person who will be great with this.
I have a question that could have implied this in the past, but I'm thinking about the profit motive. We had a lecture a few years ago about the fact that pharmaceutical companies prefer to develop medicines that you take every day for the rest of your life, as opposed to those that will fix something. Double-blind studies must be very expensive. Is there a competition for them? And if there's a competition, to what extent does this profit and profitability figure in?
Yeah, but there's a certain narrative around why are we in this antibiotic crisis because of that story. So again, if you look at the 50s, the antibiotics were the most profitable drugs. They were the recipients of the first clinical trials. And like you say, by the 60s, certainly by the 70s, 80s, you started having increasing investment in these lifetime drugs, cholesterol drugs, antihypertensives, diabetes drugs. And the narrative is that the pharmaceutical industry said, well, those are more profitable drugs because you are taking them for life. You're not just taking it for the 7 days course of pneumonia. Why don't we invest in that? And therefore, and they played some role, after antibiotics were being developed during that time. So certainly, this profit motive shapes, at the very least, which drug classes are being emphasized. Absolutely.
I just want to follow up by thanking you for a lecture that's been filled with information and provocation.
Oh, thank you. Thank you.
The questions?
Please join me in thanking again our speaker. I want to let you know we will have another lecture in 2 weeks. Adolf Reed is going to be talking to us about issues of race and healthcare. So please join us for that. Also, we'll be live and on YouTube.